This video is a presentation for the Spring 2020 Host Pathogen Interaction Journal Club at the University of Maryland. Since many of you do not know me, I'd like to briefly introduce myself. My name is Richard Zipper. I'm a retired software developer. In my youth, I received a degree in math with a minor in physics from the State University of New York at Albany. When I retired in 2011, I joined the University of Maryland as a Golden ID student, and in 2017, I received a degree in cell biology from the University of Maryland. During some of that time, I volunteered in Dr. Wozniak's lab, and I am currently a volunteer in the Aribi Center Makerspace. The paper I am reviewing is from the February 2020 Science Advances Journal. The title of the paper is A Single Nucleotide Polymorphism in Plasmoduim berghei ApiAP2 Transcription Factor Alters the Development of Host Immunity. But before I dive into the paper, I'd like to present some background regarding Plasmoduim berghei. Plasmoduim berghei is a parasite which infects mice and usually results in fatal case of anemia. It is commonly used as a model for studying malaria. Malaria is caused by parasites of the genus Plasmoduim, the deadliest of which is Plasmoduim falciparum, which is responsible for more than 400,000 deaths a year, more than half of those deaths being children under the age of 5 years old. There are still many unanswered questions regarding the mechanism of how malaria disables our immune system. As a result, there is no vaccine at this point. From a cell biology point of view, Plasmoduim has many interesting properties. Being in the phylum Apocomplexia, the cell contains an organelle in the apical part of the cell called the apicomplex, which is a plastid surrounded by 4 membranes, has its own 35K circular genome. In addition, the cell contains an apical complex which contains specialized organelles like micronemes, rope trees, and unusual cytoskeletal elements such as the conoid, all of which are used to invade another cell. Second, it has only one mitochondrion. Finally, it reproduces by a process called schizogony. Schizogony uncouples nuclear replication of the cell from cell division. After nuclear and organelle replication, the inner membrane complex engulfs the cytoplasm and the organelles create daughter cells, leaving behind a membrane with waste products. Plasmoduim typically produces 16 daughter cells in this process. I hope this background provides some context for the next video when I dive into the paper.